Yes, you're audible.
Thank you. Hello, everyone. My name is Mohit Mazumdar, and I am a bioinformatician by education. And since last 12 years, I have been practicing and applying bioinformatics for research, and now both for student-driven research projects and academic and industrial collaboration. So during my PhD, I learned to apply machine learning algorithms to variety of sequence-based data sets, and were able to design and complete several research projects that are now published in peer-reviewed journals. So with this incredible growth, I think big data has turned out to be a remarkable career choice for many today. Industries from various domains offer different positions for big data professionals. That includes biology and healthcare too. So some of the topics that we are covering in this program are quite relevant to the industry. So from a data perspective, I think we have already heard from Ilya a lot about it. So what I think is what makes biology unique from other realms is its sheer size. So I think that, for example, if I have to give you an example, a typical genome of a microbe, which might have 4,000 genes, and if we assume that there are like 1,000 meaningful distinct variants for each gene, then that's like almost 1 billion samples to test. So I mean, if you consider that sort of volume of data, it's really very big. So genomic researchers, they utilize bioinformatics to speed up processes related to analyzing DNA sequences, ultimately helping inform teams looking to combat a given disease. So COVID vaccines and cancer research are two areas of genomics that is greatly helped by bioinformatics. Similarly, in the case of drugs, as vaccines and future cancer treatments, current pharmaceuticals can also become more efficient, and they are moving in that direction with almost instant analyzation capability afforded by bioinformatics, right? And machine learning helps to make databases more searchable and trackable by biologists whose human understanding and instinct are paired with power of AI, and that power of AI can create a kind of an investigative ecosystem. So that's something that we all are looking forward to. About this program, I think our team and industry within our team and with our industry and academic collaborators contributing in developing this unique program that takes you through the OmicsLogic learning experience and provide hands-on training through the powerful eBioinfo platform. So let me tell you about the intended outcomes and some of the learning objectives of the program. So some of the things that we will be doing is that we will be learning about the terminologies for machine learning and artificial intelligence in biomedical discovery. We will be understanding the analytical methods for processing, visualizing, and analysis of complex biomedical data. We will be understanding the machine learning taxonomy and also the commonly used machine learning algorithms which are used to analyze different kind of biomedical data. We will have hands-on practice in application of standard unsupervised and supervised machine learning methods to various types of data such as genomics, transcriptomics, metagenomics, imaging data, and clinical data. You will be becoming familiar with project examples where machine learning was used effectively to achieve meaningful results. And then you will be able to understand differences between machine learning categories. Categories of algorithms in different machine learning algorithms in different categories, which would be like what kind of data sets they are good for, and what kind of problems they are good for, and they are applied to. And then we will understand about different applications of machine learning in the aspects of project design objectives. And then we will learn about Python packages for data visualization and analysis of and using analysis using machine learning methods by Python, and then apply machine learning techniques to analyze public domain data or your own data sets. So those are some of the things that are intended to come out of this program. So this course and this program is designed as a combination of online resources. Then there will be practical assignments, and there will be live sessions, live workshops that will be conducted through Zoom. And throughout this program, we will review several project examples that demonstrates the success or the limitations of conventional machine learning methods and also deep learning methods using data from public repositories. Okay, so now let's go to the sessions, that what sessions will be the next. So I would wanted to give you an overview of what is coming next. So in the next session, that will be the second session. In this session, we will learn about various omics data that are generated through high throughput technology and contains detailed information, for example, genomics, transcriptomics, metagenomics. And then we will learn about the overview of approaches, and we will learn from use cases for various types of machine learning algorithms for clustering and classification to visualize and interpret multi-dimensional data. The fourth session, in this session, we will overcome some of the challenges to compress complex information into an interpretable visual representation accurately. And in the fifth session, in this session, we will learn about the very critical challenge to organize data using complex patterns in a space that is beyond human imagination, and we will be in the multi-dimensional space, as I said. And in the sixth session, we will be learning about the application of supervised machine learning for classification. In this session, we will be learning about how do we train or training an algorithm to find and use patterns that we think are meaningful. In the seventh session, in this session, we will learn about feature selection and gene signature construction. And after that, in the next session, we will be using regression and generalized linear models to understand relationships in data by finding associations between a pattern and influence. And then in the next session, which would be the ninth session, and this would be in November, we will be learning about the system of interaction between many players, for example, across tissues, across time points, or across individuals. So after that, in the 10th session, we will be combining machine learning methods, and we will be understanding that how machine learning methods and with predictive modeling, we are going towards automation. So we will be learning up from some project examples to understand how machine learning is helping in automation. And then in the next session, we will be diving deep into the deep learning to make a network deep learning approach, which is we will learn for classification. So there are different types of deep learning methods that we will be learning about, and implementation of time series data. And then it would be about the project. So after learning and applying all these different tools that are available for a data scientist to be applied on different types of biological data, we would be working on with participants to develop a project or to be able to work on group projects or ongoing projects. So here we will be discussing about project design, understanding of the use cases for data analysis, and the need for data structuring for before you go to the analysis. And while doing this, we will be also taking you through some of the examples, and those examples are from clinic. So in the clinical condition, I'm sorry, let me go back to the previous slide. Yes, for the clinical condition, I think we would be talking about biomarkers, diagnostics, treatment, and also drug repurposing. And after that, we would be talking about the industry challenges. So we'll be talking about pharma and biotech companies and their need for machine learning and data mining projects. So overall, we will be taking you through some of the resources and some of the problems from the 20th century and learn about the importance of these technologies in the future. And we will be using tools such as R and also Python to extract meaningful insights from large biological data sets, learn, practice, and achieve biological greatness with concise exercises and interesting challenges right in front of in comfort of your own browser. So that is one of the advantages of this program. And to tell you more about the resources and the data sets and the opportunities, I would like to invite our community manager, Ms. Sonalika, and I will be happy to take any questions that you might have for me.
So we have a very consistent question coming up that what about the beginners? How will they start with the languages or let's say data science or machine learning? Because there are a lot many topics which are covered here. So could you please highlight on that?
Yeah, so actually that is being something that we discussed earlier also. So this is not just based on the live sessions, but you will have access to all the asynchronous courses. And right now, Sonalika will take you to the asynchronous courses that is actually built for anyone. So anyone from biologists to clinicians, from experts can learn from those asynchronous material. So those courses are designed in a way to make sure that you go through the content and learn about what we will be covering in the sessions too. So I think if you start going through that material, which is available online, and you can do it from actually anywhere, I think that would be really helpful for you to go through the sessions and interact with our mentors and design a project. So all of this is possible when you start from if you are starting from scratch, and that is from the asynchronous material on machine learning, on different type of omics data, on different type of application projects. Everything is available on Learn OmicsLogic, and I think Sonalika will be able to show you how you can get started there.
Thank you so much, sir. If there are any other questions, you can post in the chat box and